A huge thank you to Nicholas and to your parents and family. We really appreciate you being here today, and you inspire us and make us clear how urgent it is for us to continue. And as Dr. Glade-Bender said, your voice is so important because it will actually help with the process of accelerating. So thank you very, very much on behalf of all of us. It's an honor now to introduce our next speaker. We're targeting lots of difficult things today. So this is another tumor that Dr. George Annor, who works at the Pediatric Oncology branch with Dr. Jack Shern, is working on. It's called malignant peripheral nerve sheath tumor. And he has some very exciting data that he will be talking about today. DLK1 as a therapeutic target in malignant peripheral nerve sheath tumors, and its expression can be modified by epigenetic modifications. Welcome, Dr. Annor.
Thank you very much. This is a tough act to follow. Nicholas, we are all wishing you the very best. So thank you, CCF, for this opportunity. Today, I'll be sharing with you work that we are doing in the Shern lab, targeting MPNST with an antibody drug conjugate. I don't have any financial disclosure. Malignant peripheral nerve sheath tumor, or MPNST, is an aggressive soft tissue sarcoma with a very low 5-year survival rate, less than 30%. Surgical resection of localized tumor is the current curative therapy, and the MPNST tumor tend to be highly metastatic and resistant to chemotherapy and radiotherapy. This therefore means that we need to devise an improved treatment regimen if we are going to improve the survival of patients that have this condition. A subtype of MPNST originates from mutation in a tumor suppressor called neurofibromin. When this mutation occurs, it leads to the formation of plexiform neurofibroma, and these plexiform neurofibromas transform eventually into MPNST with the loss of function of other genes. One of the genes that gets mutated is polycomb repressive complex 2, which is a stop signal during gene expression. In a normal cell, a functioning polycomb repressive complex is able to prevent the transcription of certain gene or put a stop on it. If we have MPNST with mutations in PRC2, this stop signal is lost and it leads to transcription of a lot of genes. So if you consider a cell, there are some of these proteins that are found on the surface of the cell. But when you have PRC2 loss, there are more proteins that are also found on this cell surface, which got us thinking that if we are increasing the level of these proteins on the surface of the cell, then we can target these proteins with a drug or an antibody conjugate. So to do this, we first screen MPNST tumor and MPNST cell line and compare gene expression that are found on the surface of the membrane to normal cells. And we found out that 33 of these genes were overexpressed. And those genes that were overexpressed, one of them was of interest to us. That is DLK1. And the DLK1, we saw that it was differentially expressed in the MPNST tumor as well as the MPNST cell line. When we use single-cell RNA sequencing to compare the cluster of cells that express DLK1, we saw that from the MPNST tumor cell cluster, a lot of them had DLK1, which confirmed that we can find DLK1 expression in MPNST tumor. So one will ask, what is DLK1? DLK1 is known as delta-like non-canonical Notch ligand 1, which is a transmembrane protein that is highly expressed during embryogenesis. But the expression becomes limited in adults and restricted to neuroendocrine glands. It's been found out that in certain cancers, particularly neuroblastoma and adrenocortical carcinomas, the levels of DLK1 is very high, which makes us think then we can target this particular protein. So to target this protein, we collaborated with ADC Therapeutic that have made an antibody drug conjugate, which you can picture as a smart bomb or a guided bomb, which has the bomb attached to a signal that is able to direct the bomb to the exact target. And when it gets to the target, it enters the target, gets released, and it detonates in the target, thereby killing the cancer cells that highly express DLK1. So we wanted to test this antibody drug conjugate, which is called ADCT701. So first, we looked at the expression of DLK1 in MPNST cell line compared to a known cell line that highly expressed DLK1. We found out that there was increase in DLK1 expression at the mRNA level in a differential manner, but the expression we couldn't detect when we ran a blot and compared it to the positive control, H295R. So I ran another gel without this positive control, and then I was able to detect that DLK1 was expressed. Now, to test the effect of treating these cells with the antibody drug conjugate, we used an assay called viability assay, and it's a cell titer glow that we use for this experiment. And we looked at how the cells responded when we gave them either the antibody drug conjugate, which is the guided bomb, or just the bomb attached to something that is not specific, or the control, B12. We saw that in the cell line that had high DLK1 expression, we required a very small amount of the drug to kill 50% of the cell, which was very promising. But in the MPNST cell lines that didn't express high DLK1, we couldn't see this in both the drug and the drug control. Thus, we overexpressed or made more of the DLK1 in the cell line. And when we did that, we were able to see that now we require a small amount of the drug to kill 50% of the cell. This was very good. Then we moved to work in vivo. And the first thing that we did was look at the DLK1 expression in 40 MPNST patient sample. We used an experiment called immunohistochemistry to do this. And we stratified the expression of DLK1 into weak, medium, and high DLK1 expression. And we use Western blots to confirm this expression in some of these tumor models. We found out that 2 of them had very high DLK1 expression, 2 relatively high, and 1 very low. But in order to see the medium expression, I ran the gel separately, and I could see that, yes, we could detect DLK1 being expressed as well. So then we took these tumors, minced them, and then we implanted them in an immunodeficient mouse called NSG. And we implanted the tumor fragment, allowed the tumor to grow to a volume of 100 millimeter cube to 150 millimeter cube, and gave a single dose of either the antibody drug conjugate, the B12, that's the drug control, or just normal saline, and measured how they responded to this particular treatment. So for the first one, Wu356, we observed that after the drug administration, the tumor volume started going down day 10 post-drug treatment, and it went down until we reached between day 60 and day 70, where one of the tumor relapsed and got to the endpoint tumor volume of 2.0 centimeter cube. So we resected this particular tumor and allowed the experiment to continue. However, in the mice that we gave the drug control or normal saline, they didn't respond, and they reached the endpoint tumor volume of 2 centimeter cube around day 30. We also looked at the survival rate after the treatment, and we saw improved survival in the mice that we gave the antibody drug conjugate. We repeated this experiment in JH202, and we saw a similar trend in post-drug treatment. Here, around day 70, we didn't resect the tumor. What we did was we gave a second dose to the 3 mice that relapsed, and now we are monitoring them to see if they will respond to the second treatment. We also looked at the probability of survival, and we found out that they had an improved survival, suggesting that if you have MPNST that express DLK1, you can target it with the antibody drug conjugate. But as I had shown that the DLK1 expression wasn't high in the MPNST cell, and it didn't give me a good amount required to kill 50% of the cell, or IC50, we wanted to understand how to regulate this expression so that we can improve the response to the antibody drug conjugate. We are thinking that the PRC2 is in part regulating DLK1 expression, as well as DLK1 is an imprinted gene, which just means that it is silenced on the maternal allele but expressed by the paternal allele, and this expression is mediated by DNA methylation. So here, we wanted to see how the PRC2 complex, which is the on-off transcriptional switch, regulate DLK1 expression. So we looked at 3 main marks: acetylation and methylation of 2 different histone marks. The first one, we compared acetylation at lysine 27 in a cell line that highly expressed DLK1, which is a neuroblastoma cell line, to a low DLK1 expression cell line. And we compared it to MPNST that has PRC2 reintroduced, or an MPNST that has mutant PRC2. And we saw increased signal intensity for the acetylation mark in the high DLK1, and a similar trend was seen even in the PRC2 mutant. With the repressive signal or off switch, which is the histone trimethylation at lysine 27, we saw that in the line that highly expressed DLK1, you don't see any significant peak, but in the low DLK1 expression, you see high peak. A similar trend was also observed in our MPNST, PRC2 wild type, and PRC2 mutant. Then the last one was improved promoter activity, which is methylation at lysine 4. We also see increased peak in the DLK1 high, low peak in the DLK1 low, and in our PRC2 sample, we see that in the PRC2 wild type, there is a low signal. This observation translated into high DLK1 expression in both the DLK1 high and our PRC2 mutant line. But in the MPNST, this high DLK1 expression was not enough to respond to the antibody drug conjugate. So the next thing that we looked at is how does methylation affect the expression of DLK1. So we treated PRC2 minus or PRC2 mutant MPNST cell line with a demethylating agent in combination or as a single dose with an HDAC inhibitor, since we saw that the neuroblastoma that made a lot of DLK1 had high acetylation. We found out that there was increase in DLK1 expression both at the mRNA level and at the protein level when you add the demethylating agent, as well as adding the demethylating agent to the HDAC inhibitor, suggesting that we can increase DLK1 expression in MPNST that have mutant PRC2 by using a demethylating agent. So in conclusion, I was able to show that DLK1 is highly expressed in MPNST tumor, and that we can target MPNST tumor that express DLK1 with an antibody drug conjugate. And this treatment improved their survival and reduced tumor volume. Also, DLK1 expression could be increased via epigenetic modification. In the future, we would like to test the antibody drug conjugate in a lot of MPNST tumor models to see how robust this particular observation is, since we think we had some promising data. We will also want to understand the mechanism that is driving the relapse that we saw in our MPNST tumor model. Since we also saw upregulation in the MPN and DLK1 expression with the epigenetic modifiers, we want to understand mechanistically how is this particular event happening. Lastly, with the increase in DLK1 in the presence of the demethylating agent, we would want to test the durability of the antibody drug conjugate in response to a pretreatment with the demethylating agent prior to giving the mice the antibody drug conjugate. I would like to thank members of the Shen Lab, those that are here and those that are at the NCI, and also Dr. Angela Herby, who has provided us with the MPNST tumor model. Thank you very much.
Wonderful. Thank you very much. Questions?
Terrific talk. I've recently learned that CARs made with T-cell receptors rather than with antibodies are very sensitive and can kill tumor cells with, say, 10 molecules of the target molecule expressed. Do you think that might be a solution for your low DLK1 tumors?
We would like to test, thank you for the question. We would like to test that. So our collaborator is trying to design CAR T cells specifically for DLK1. And the goal is when we get it, we will test it in our low expressing DLK1 as well as those that relapsed.
You might have to do it as a T-cell receptor.
Yes.
Maybe I can ask one question. There is a clinical trial ongoing with this now. Do you think it will be enrolling patients with MPNST, and what would you want to get from patients that enroll on potential trial as samples?
Thank you. Soon we will be starting clinical trial. DLK1 ADC701 is actually in clinical trials now, but in neuroblastoma. So with this data that we have, we are trying to expand in other MPNST tumor model, and then we will start. I think Jack has already started pressuring us to start working hard towards getting it in clinical trials. And the second part of the question, we would want to get the patient tumor sample to also understand what is happening to those that respond to the antibody drug conjugate, as well as any relapse that occurs. So we will get it at different time points of the treatment. But the interesting or the good thing is it is a single dose treatment. So if it is working effectively, you wouldn't have to come in every time to get the treatment.
Are there any other questions? If there are no questions, thank you so much for, oh, sorry, Julia.
Just since it's the question of the day, what would you think to combine? What would you think to combine this therapy with, knowing that it most likely won't work as a single agent?
I think I'll look into combining with a DNA damaging agent to improve it.
Also, the azacitidine.
Yes, so that one, we are thinking the dosing that we will use, it will be to increase the DLK1 expression. But it's possible for us to also use it as a combination therapy. Yeah. So thank you all very much for this opportunity once again.
Thank you all. I think we have a break now.